Okay, I'm recording. So I'm Kelly Ruggles. I was a postdoc with David, and I'm still working with him quite a bit. Doing one of the projects that we're working on has to do with this actual topic. So I'm going to go through protein quantitation, the second part, looking at multiple reaction monitoring, and of course just ask me questions as we go if you have any. So just a general outline. I'm going to go through an overview of protein quantitation using MS-MS, then move into the basics of targeted proteomics, and then use a motivating example of AKT in breast cancer to sort of work through the workflow of targeted proteomics. So starting with the protein quantitation overview, some of this you've probably covered, but I want to get us sort of all on the same page and give some context to the targeted proteomics. So proteomics has been described as the quantitative analysis of proteins expressed in a cell at a certain time. And so you can think about this as either looking at all of the components of the proteome. So this is the global analysis where you complete a one-time look at all of the proteins within the proteome using mass spec. This is also known as discovery proteomics. And the majority of proteomics papers have used this technique. And the second type of proteomics is looking at dynamic changes in the proteome. So this is more like a clinical or a biological assay where you are looking at the proteome in under different conditions or through time and maybe focusing on one or two proteins instead of all of the proteins in the organism. So we'll just start with looking at all of the components of the proteome. So this is discovery proteomics, which I'm sure that David has talked to you about quite a bit. This is just the general workflow where we have a protein sample and we do an enzymatic digestion, get our peptide mixture, and do liquid chromatography tandem mass spec. We get our spectra, and we use the spectra then to match two peptide sequences. So this is the first step in the computational analysis. We then use these peptides to map back to the protein sequence. So there's a couple of steps of computational analysis here, and some of the issues within these computational steps are, first, of course, the peptide identification, so using the databases in order to match the spectra to the actual peptide sequence. And then the second is called protein inference, where there's the issues going from the peptide to the actual protein. And I'm going to go into a bit more detail about this. This step has been really well studied, and it's pretty robust at this point. So let's focus a bit on the protein inference. So as I mentioned, there are some computational issues specifically with protein inference. So in order to generate a reliable list of proteins from the identified peptides, this is not a very straightforward process. And there's things called one-hit wonders, where a protein will only have a single peptide identified. And because of false positives, it's really difficult to say that that one peptide means that that protein is there, right? So, and also, multiple proteins can be supported by one peptide, and determining whether that peptide belongs to protein 1 or protein 2 is difficult. So here's just an example, a schematic. Here we have our peptides in yellow and our proteins in green. So proteins 1 and 2 have the same set of peptides 1 and 2. And if we don't have any other supporting information, then we can't figure out whether or not peptides 1 and 2 correspond just to 1, just to 2, or to both. So that's one of the first sort of issues with protein inference. The second is that we have a one-hit wonder here where peptide 3 is the only peptide that was found for protein 3.
Is that called like the unique?
So it's a unique, yeah. So there's unique, that's the only unique peptide that was found, right? So this one's unique. 1 and 2 are not unique, right? So we can't necessarily say that we have found protein 3 if we only have that one peptide. And here we have protein 4 where we have two unique peptides 4 and 5, and we can say, great, 4 is there. So there are some issues if you want to know, if you really are interested only in protein 3 and you use this type of analysis, it's hard to say that you've found protein 3, essentially. So there's been a lot done to draft a map of the human proteome. Here are two papers that were recently published looking at the entire human proteome using this discovery style of mass spectrometry. So these methods are robust, but there are still some issues like the protein inference we just talked about, also reproducibility and false discovery rate. So for people who are more interested in clinical and biological assays, this might not be the best approach, right? If we want to look, this is looking at all the proteins in the organism, and if you want to know about 3, maybe this is not the best way to do it, right? So instead, we need to focus on a subset of proteins and ensure that we can achieve reliable quantitation of those proteins. So that's where we get to the second part of protein quantitation, looking at the dynamic changes in the proteome and focusing maybe instead on a couple of proteins and looking at them either many times or across many different conditions. So traditionally, this is done using antibodies. Anybody who's worked in a wet lab, I'm sure, has done a Western blot. I know I did a million in my PhD. So here are just a list of different antibody-based assays, so Western blots, ELISAs, reverse phase protein arrays. So these are ways that have been traditionally used to look at specific proteins of interest. And most of the clinical and biological studies that have been published in the literature have relied on proteins that already have antibodies, right? So the goal of targeted proteomics is instead to create a reliable, high-quality assays that can measure proteins but not require antibodies. Because if you've worked with antibodies, you know that some of them, they either go bad, they aren't good, they're not specific. There's all sorts of problems with that, and they can be really expensive. So instead, if we can rely on tandem mass spectrometry, that may be a better approach to quantitatively measure proteins in the cell. And this method was considered the method of the year by Nature in 2012. And it focuses, again, on a subset of proteins of interest. So you can look at disease-related changes in proteins, signaling processing, and you can use highly multiplexed methods instead. So you can do, like hundreds of proteins and do really specific quantitation instead of doing hundreds of Western blots. So it can be a really powerful method. And it's instead of this sort of global proteomics, you can do more hypothesis-driven questions. So this is just sort of a summary of when to use targeted mass spec versus global shotgun. So for shotgun mass spectrometry, again, we identify the maximum number of proteins in a limited number of conditions. So this plot just shows the protein expression and RNA expression in one experiment, and the regression showing the correlation between those two, right? So if that's what you're interested in, you have to do a global proteomics experiment. Instead, if you want to look at a limited number of proteins in the maximum number of conditions, so here we have a certain number of proteins in 4 different types of breast cancer, then targeted mass spec is the way to go, essentially. So now we're just going to go through some of the basics of targeted proteomics. And just to compare the two on a little bit more of a detailed level, here we have our typical mass spec workflow, where we have our cell, we lyse the cell and fractionate, enzymatically digest it, and put it through the liquid chromatography mass spectrometry. So in shotgun proteomics, it records the M over Z and then selects the peptide based on the abundance. So it will select the highest abundant peptide here, and then it will fragment that, and then that will then be collected as data. So essentially, you're getting the highest peptides that have the highest abundance, and you're fragmenting those in the tandem mass spec.
What if you're interested in the low expressed peptide?
So if you're interested in the low expressed peptide, you almost certainly have to do targeted mass spec, because what happens in targeted, so if we go through this, we can actually tell the mass spectrometer, I'm looking for this peptide. This is exactly where it's going to be. Only fractionate that. So it will pick out, let's say it was this one. Let's say this is actually, this is only one peptide, but let's say it was really low. It would pick out that one, and it would just fractionate that. So as long as it is in your sample to like a reasonable degree, you would be able to see it. If it's not there at all, you're not going to see it, right? But
when you pre-select, like isolate those proteins of interest, what if there is a similar peptide has the same M divided by Z?
yeah, that is a huge problem, and we're gonna talk all about that.
Oh.
Yeah, yeah. So, but that is exactly why you would do something like this if you have either a low expressing peptide or you really want to be like specific about what you're looking for. You would do targeted mass spectrometry and be able to say this is what I want. So there are a couple of different kinds of targeted mass spec. The first is selected or multiple reaction monitoring. These are the same thing. There's just some people call it SRM, some people call it MRM. I'm going to call it MRM here. And then SWATH-MS spectrometry, which we'll also talk about just a little bit at the end of the lecture. Okay, so multiple reaction monitoring. So here I'm going to go through a little bit about the actual mass spec. It uses a triple quadrupole, and here's what a normal mass spec experiment looks like, where you have your peptide, it fragments whatever the highest abundant peptides are, and it detects all of those fragment ions. In our targeted, we select here our peptide. We then fragment only that peptide, and then we select for our fragments. So we select twice. We know what our fragments are going to look like. We tell the mass spec, I want you to select for this peptide, and I want you to select for these fragments, and I want you to just read at the end what that looks like. So there's two steps of filtering here, essentially. And this is, again, just what this instrument looks like. We have the mass filter here, which selects for the peptide, the collision cell, and then it selects again for the fragment ions in the third, in Q3. Okay, so this is just showing a bit of, so these are the three different fragment ions that we selected for, Y6, Y7, and Y8. And this is just sort of showing where that fragmentation occurred. So for Y8 it would be here, for Y7 here, for Y6 here. So we're actually collecting data on these three different fragments of the peptide for light and heavy. So that's just sort of what the end spectra would look like. Okay, so in order to then identify your peptide, you use both the precursor mass and the fragment ion mass. So that pair is used in protein identification, if that makes sense. So you know what your M over Z here is, you know what your M over Z here is, and then together that's called a transition. And those are used to identify your peptide. And you should know that before you actually do the experiment. And usually when picking the transitions that you're going to select for here, people choose the three most intense that either when they've done an experiment to see what the transitions are when you actually fragment this, or there have been a lot of experiments that have been done where there's a lot of resources where you can guess what the three most intense transitions are, and we're going to talk a bit more about that also. So this was actually originally used to analyze small molecules. So it's just recently that people have actually used it in proteomics, and particularly for biomarker discovery. So with the small molecules, the matrix that they were using and the actual small molecule were really different from each other. So separating out the small molecule from the matrix was really easy, and they didn't have to worry about like getting any of this interference from the matrix in their actual analysis. But now with proteomics, both the proteins of interest and the matrix that they're in are really similar. So we do have to worry a bit more about not separating those two out. And I think this gets at the question that you brought up before. So, and we're going to go into a whole lot of detail about that. But the strengths of MRM are that they can detect multiple transitions and analyze many peptides at once. So instead of, like I said, instead of doing 100 Western blots, you could do 100 MRM experiments.
I'm sorry.
Oh, it's okay.
I just don't really think I understand the transitions. How to
Yeah, so the word think about the word transition as a fragment ion. And so it's really another, and that is another way of people saying it. So the actual fragment is the transition. So the fragment and the precursor together are considered a transition. But just think about it as the like fragment of interest, a fragment ion of interest. Yeah. So when I say like many transitions per peptide, that means that like you can look at 3 of the fragment ions for every peptide you put in. So you fragment a peptide, and then you can select for 3 peptides, 3 transitions, and then record those, essentially. Does that make sense? Okay. So it's really sensitive and reproducible, and it's able to detect low levels even in pretty complex matrices. And right now it's considered the golden standard for quantitation. So there are some weaknesses, however. So it focuses on a specific set of peptides, right? So you have to tell the mass spec, this is the peptide I want. So you need to know a lot about that peptide, including the charge state, retention time, the ion intensity before you start the experiment. Instead of just throwing your sample in and like seeing what happens, you have to do a bit more research beforehand. And there's a physical limit to the number of transitions or fragment ions that you can collect in that last part of in that second step of the mass spec, right? So right now it's typically 3 in an MRM, but you can get around this by using time-scheduled MRMs, and then there's a thing called parallel reaction monitoring that we're going to talk about in more detail as well. But that's one of the limits to MRM. So leads into my parallel reaction monitoring. So in parallel reaction monitoring, so it's SRM, remember, we choose our peptide in the Q1, we fractionate that peptide, and then we choose our transition in Q3. And then we record the intensity of that. So in parallel reaction monitoring, instead, the Q3 has been replaced by a high-resolution mass analyzer. So it's able to choose here, fractionate, and then look at everything that it fractionated. Instead of just choosing one, it can look at all of this. And then we can say if you had a peptide that had 12 possible transitions, you can look at all 12 of those, which is really powerful. And then also you don't have to choose these ions beforehand. So, and this is just what, for those people who like to look at instruments, this is just what that looks like. So instead, this last step here is actually a time of flight mass analyzer. So it's a lot more sensitive than the MRM, that last readout in the MRM. So there are a lot of applications of MRM and PRM. The first is looking at metabolic pathways. So so if you are if you're looking at a cell and you want to know like all of the things that are happening in a specific pathway, you can actually look at all of those if you do an MRM for each of those different proteins. The protein complex stoichiometry, same kind of thing. You can quantitate exactly how much of each part of your complex you have. Phosphorylation levels, so you can actually do phosphopeptide MRM. Any kind of modifications similarly within a protein. And then again, the most commonly used thing is biomarkers. So in cancer or any other disease, looking at the difference between a level of protein in a tumor versus a normal cell. So I'm just going to use a motivating example to go through the process of how this sort of works. So just for simplification, I'm just going to do AKT and breast cancer. We're not going to go through any of the details of this pathway, but just, as an example, we would maybe pick 10 of these and look at it in a whole subset of breast tumors and see how those vary across different tumors and how they differ from normal tissue, right? So let's say that's our question. So this is the MRM workflow. We define a protein set based on whatever our question is. From this protein set, we then select peptides. From the peptides, we select transitions. And then we have to test the transitions, make sure they look good and we're getting them and whatever. And then we validate these, and then we can do our quantitation. So I'll go through each of these steps one by one. So first, define our protein set. Again, let's just say we were interested in this pathway in breast cancer. We would pick, let's say, the AKTs and maybe a couple of other proteins of interest and then from those we would select those proteins, and then we would select peptides within each of those proteins. And that's a bit of a longer step. So we want to pick a few representative peptides, because you don't want to just use one. You want to use like at least 3. And they have to fulfill certain characteristics. So they need to be unique, obviously, because if they're not unique, who knows what you're measuring. They need to be consistently observed by other mass spec methods. They need to be, of course, within the length that a mass spec typically measures. So let's say 8 to 25 amino acids. They need to have a good ionization efficiency, an M/Z within the range of the instrument, typically no miscleavages, and then not too hydrophilic or hydrophobic. So those are just typically things that you look for when you're looking for like a peptide that will quote unquote fly in the mass spec. So ways of identifying unique or proteotypic peptides. So let's say we have a set of proteins. Here's our FASTA format sequence. We can do an in silico digestion, so just cut it where trypsin would cut it and look at that list of tryptic peptides. And then compare these peptides to all of the human reference databases and just make sure that they only match to our protein of interest, essentially. And then, so you first match them to proteins and then match them back to genes. So if they all match to your gene, then you're good to go. And some proteins, unfortunately, like the AKTs, actually have very few proteotypic peptides because they're all very similar. So you have to be like really careful which ones you pick. You can also use a thing called GPMDB. Has David talked about this at all? Okay. He loves it. So, where you can put your peptide in, and then it shows you. So the more red it is, the more frequently it's been seen. If it's green, that means it's probably not gonna show up. So this is a good check to make sure that you're gonna pick the right peptide, because most proteins show up in very reproducible patterns. And then Skyline. Have you guys heard about Skyline at all yet? I know that you're going to, next week, I think you're going to get like a whole thing on, or not next week, 2 weeks from now, you're going to get a whole thing on Skyline. But it's like a very commonly used tool for mass spec. So you can put, it's kind of hard to see here, but each of these is a peptide sequence. And like you can click on it and it will show you all of the transitions that you're most likely to see in that sequence. So it's a good tool to see like what peptides mostly show up and what transitions within those peptides also show up.
I'm just curious.
Yeah.
Do you think that kind of software is going to be interference? Like we have a set of peptides and people tend to use the similar peptides as the standard. So it's very naturally more and more people select those kind of peptides.
Yeah, a lot of this data is actually from discovery proteomics, though. So I know what you're saying, but like, because this isn't, a lot of this stuff is not targeted. So you're using data from non-targeted and then looking to make sure that peptide shows up when you just don't select for anything, when you're just like letting everything come through. Because even though you're targeting, you want to make sure like somebody has seen it at some point in time. Otherwise, it's very unlikely that you're ever going to find it. So that's usually why this is like, it's not the only resource, but it's good to like check. But it's similar to GPMDB in that like it's data, a lot of data from other experiments, but it's similar. Okay, so after we select our quality peptides, then we have to select our transitions. So one of the limitations, again, that we've sort of talked about is that occasionally that we can let in interfering peptides. Maybe we didn't talk about this that much yet, but where if it has similar M/Z, so let's say you have your precursor ion and you have another peptide that's almost exactly the same, right? It's going to come in, it's going to be fractionated. Then you have another, it has a similar transition to the one you're looking at. That's going to get in also. So you have to be really careful because you're filtering twice, and it's possible that you could get things that come into both of those filters, right? So we need to be really confident that we don't have interferences. So the way that people have dealt with this is thinking like, okay, if our precursors for this peptide, the largest is always largest, the smallest is always smallest. So it's essentially like if the Y5 is always the smallest, it should always be the smallest. And the Y7, we're going to go through this in more detail, but like looking at proportions of how your transitions look later. So I'm sorry that this like is the formatting got messed up. But here's just an example of what, okay, so let's say our peptide of interest is yellow, and this is what our MS2 spectra look like. And we have an interfering peptide that's gotten in, and its transitions are in red. So if that's the case, we're seeing a much higher intensity for some of these transitions than are actually true, right? So there are a couple of ways that you can do this before you even touch the mass spec. This is one program called SRM Collider where you input your peptide sequence and you put in all sorts of different parameters that have to do with your mass spec. And it simulates a SRM or MRM experiment where it predicts the fragment intensities and retention times. And then it compares the transitions that it thinks are going to occur to all of the other transitions in their sort of like database and outputs what they think is going to happen. Of course, this is all, a little bit, because it's not a real experiment, it's hard to say that this is exactly what it would look like, but it is one way of sort of looking through it beforehand and making sure that you're not going to see some crazy amount of interference. So the other way is, again, Skyline, where you can pick your peptide and then it actually tells you, based on a dot product, whether or not, so it looks at the intensity, and also a dot product close to 1 means that you're less likely to have an interference. So this is just another way of them like taking the data they have and using it to tell you what they think is going to be in your data. So those are 2 ways to sort of do it beforehand, but they're not foolproof. So usually what happens is you select your transitions, you actually run the experiment, and then you have to validate them and make sure that you're not seeing interference. There are a couple of ways of doing this. One is called a branching ratio. So you have a light and a heavy peptide. So typically these are all done, and I'll go through more about this, but I think you've probably gone through like using heavy to quantitate. Yeah, perfect. So if you have your light analyte and then your heavy peptide, you're going to essentially select for the precursor for both of those, fractionate them, and then look at their transitions. And that's when I was saying the largest should be largest, smallest should be smallest. You're comparing the heavy and light, right? So here, your first ion or your first transition, right? Ion 1, ion 2, they should sort of look the same, and they don't here, right? So it's likely that maybe let's say there's an interference in I3 in the heavy, because you would expect I3 to be below I2. So people use a branching ratio, which I'll go through a bit, to actually just have an automated way of saying that, essentially.
What if during your fragmentation, the 2 daughter ions has 1 neutral ion? Then you don't have anything to as a reference.
So you're saying if one of them is a neutral ion in your fragment,
one return all the charges and the other one is neutral, then you don't have anything.
Then you've done a bad job choosing transitions, right? Yeah, I mean, at that point you'll notice that, and I think you'll just have to pick a different transition. Okay. Yeah, but I think it should be. I think you'd be able to like predict that beforehand, hopefully. So I don't think that happens very often, but I think you just would have to start over. Yeah. So there's another way of doing this called AUDIT. This was developed at the Broad, and they actually use this branching ratio to just automate. So they essentially like look at the relative ratios between heavy and light, and because through that they're able to say there is probably an interference in this ion. So this is just an example. This first panel A, the heavy is red, the blue is light. So they just looked at the proportions, and you can see they look really pretty good for this peptide, right, at each of these different concentrations. So audit says that there are no interferences for this peptide. For this peptide, however, so we have 3 molar of this analyte, and then you 50 sorry, femtomolo of the heavy, and we can see that they do not look anything alike, right? So audit would pick out Y5 here, the blue, as having an interference, because that's the reason why the pattern has been thrown off, essentially. Can you see that? That's okay. So that's essentially how audit and most of the interference algorithms work, using these proportions, typically through looking at heavy versus light. So I mentioned this parallel reaction monitoring before, where you're able to, instead of choosing a transition, you can fractionate everything in your peptide and look at all of the transitions. So we're able to skip this step here. So in order to do this, we've actually created an algorithm specifically looking at parallel reaction monitoring, where we're looking at all of the ions that come from your peptide. So PRM and MRM are most useful when we're looking at a complex matrix, right? When we're doing the kind of experiment like this, most of the time we're looking at like tumor lysate or blood plasma, right? That's what most people who are working in proteomics, they're not working in buffer, right? So what we and our collaborators have done are use tumor lysate as sort of a reference matrix, and use use buffer, not tumor lysate, use buffer as a reference, right? Because it shouldn't have a lot of like random peptides floating around. But tumor lysate will have a lot more likelihood that it's going to get into your filters. There's going to be more peptides that are going to make it more likely to pass through both of your Q1 and Q3. So when you're selecting for a precursor, there's more. If you're working in tumor lysate, there's probably going to be a peptide that's going to look a lot like whatever peptide you care about. And then when you fractionate that, there's a higher likelihood that you're going to get a transition or a fraction ion that's going to look a whole lot like the ones you care about, right? So we have done something where we're doing both the experiment in simple and the experiment in a complex matrix, and then we're using those two to figure out where the interferences are. So I'm going to go through that a little bit as well. I'm going to go make these bigger because they're hard to see, but this is the general workflow. So again, we have our reference matrix. So this is our it's like a buffer matrix, and then we have a more complex matrix, which would be like a tumor lysate. So let's say this is our spectra for our peptide of interest. Say it's AKT1, and we're looking just at these 3. In PRM, we'd be looking at all of these, but I'm just going to use these 3 as an example. And these is just the intensity of these 3 transitions in reference and complex. So we make a branching ratio, and a branching ratio is just essentially all of the different ratios that you can, the permutations of ratios for all of the different transitions. So of course, along the diagonal, there's going to be 1, so we don't care about that. But here, Y5 over Y4, so we get all of these. And so since we're looking at our reference, this is our reference, we assume there are no interferences in our reference because it has very little peptides that could sneak through our filters, essentially. And what we can say then is if we were to look at the ratio of our ratios in reference to our ratios in complex, they should be 1, right? So if there is no interference, these should look very similar. So Y5 over Y4 should look very similar in our complex matrix as it does in our reference matrix if there's nothing in the complex matrix that's like throwing off the ratio. So if we had an increased number in Y6, let's say, then our complex would be higher, which is actually what we're seeing here. It looks nothing like our reference. Does that make sense? It's a little bit complicated. So all we do is we just actually divide the 2 and look for things that don't look like 1. And through that, we're able to then identify Y6 as having an interference. So usually this would be a much larger matrix. That's why we automate it, because nobody wants to look through all of these numbers. But that's just another way. Instead of looking heavy versus light, we're looking at complex matrix versus reference simple matrix. And then we can, we've built something in where we can actually rank the transitions from like best, so least interference, to most interference. And again, Y6 gets sort of flagged here as having a lot of interference. So that's the general workflow and differences between MRM and PRM in terms of a more computational and informatics aspect. From here, once you have the transitions that you like, you think they're good, you think they don't have interference, you can actually quantitate your protein. So in order to do this, you have to use at least 3 transitions. So you can either do a label-free quantitation, where you actually rely on the mass spec signal. And this is really easy, but most people don't do it this way. Most people use a heavy label. So this is what most experiments rely on in targeted mass specs, so stable isotope dilution, where we use an isotopically labeled reference protein, either a 13C or a 15N labeled peptide. That's exactly the same as our target peptide, but just with the mass difference. And then we can compare heavy versus light, and since we know how much heavy we have, we can sort of determine how much light we have, right? So the way we would do this we would find a peak area ratio. So we would do the peak area of the light, which is what we want to know, over the peak area of the heavy. And then in order to get the analyte concentration, we would just multiply by whatever heavy we had thrown in. So that is how the quantitation actually is done. So there are a whole lot of other open source tools that have been created to do this. I'm obviously not going to go through all of this, but this is in the world of targeted proteomics. Interference is a huge deal, and it's something that people have spent a lot of time working on, and I think people will continue to work on, because I think this will become a bigger deal with time. So the only other thing I'll just touch on really quickly is called SWATH. So SWATH is similar to MRM in that it's targeted, but it uses a slightly different instrument. So it's a quadrupole time of flight high resolution instrument. So instead of choosing your precursor ions, it actually just fragments precursor ions within different windows. So it will fragment all of them and then record the fragment ion spectra. So it's sort of more similar to discovery based, but it's actually building in the fragmentation and the data collection that are used in MRM. So you're getting a little bit more of a messier data set, but you're getting a lot more information. And I think with time, this is a pretty new method, but I think as we get better at actually doing the informatics afterwards to figure out exactly what's happening, it will become a much more used method. So I'm actually not going to go through that, but here's just looking at interferences. So here is MRM, and the black bars here are peptides with less than 4 interference-free transitions, and the white is peptides with 5 or more. So the more white we have, the less interference. So here in MRM, we have very few. It looks like about 5%. And with SWATH, that gets a little bit worse. But as we low resolution instruments, we get quite a bit more. And if we don't have an isolation window, we get a huge amount. So this is just showing the huge variation between the number of interferences that occur across different methodologies. So I'll just summarize, and then if anyone has any questions, let me know. So from this, I've shown that targeted proteomics can be used as an alternative to antibody-based protein assays. And it allows for multiplexed analysis, so many different proteins and many different conditions, especially once you've already found a protein and transition combination that work. You can just repeat it on a whole bunch of different things. So once you get all of the upfront work done, it's really easy to repeat. And parallel reaction monitoring removes the step of transition validation, and it allows for all of our computational analysis to happen after we've actually done the mass spectrometry. And then we talked a little bit about SWATH, which removes that first step of peptide selection, where it allows us to generate transitions for all of the different precursor ions in the precursor window. So PRM first removed that last step where we had to select the fragment ions, and then SWATH removes that first step. And then transition interference must be appropriately identified. That's a really huge deal because otherwise the protein quantitation is going to be completely wrong. And several computational tools to predict and identify the interferences have been developed and I think will continue to be developed. It's a big need within the computational and informatics world. So with that, if you guys have any questions.
So, as a disadvantage or advantages, what is the advantage of PRM over MRM?
So the big advantage for PRM, and I probably didn't make this a big enough deal, but if you look here, so this step can be really annoying and really long, and if you don't have to do that step, it takes a lot. You don't have to like try a couple of transitions and then realize they don't work and then pick another, so you can just look at all of them and deal with it later. And I think that's the major advantage.
As a disadvantage, is there any?
It's a little bit, I think that it's a little bit harder to analyze the data, I think, but not enough that I think it will be a huge barrier. Yeah, I think this has become a much more popular, PRM is becoming much more popular now that those instruments have been developed. But these instruments are so expensive that it takes somebody buying that instrument to then be able to do it. So I think that will be the biggest barrier. Yeah. Any other questions? Okay. Great. Let me go from